Hello and welcome to episode 9 of Bite-Sized Biocompatibility. We're here to talk about systemic toxicity this time.
One of my favorite topics.
Yes.
I have to add, I mean, this is definitely one of the most, as we'll discuss, value add, in my opinion, aspects of medical device testing, potential for great data and risk mitigation.
And really having a look at things like synergistic effects as well. But before we dive right into it, I just want to give a quick warning that we do talk about animal testing in detail with this episode. So for anybody who maybe feels a little bit more strongly about that, please just be aware that there are some hard topics that we cover.
It is an unfortunate aspect of this type of testing and endpoint, or effect. So yes, caution to viewers.
Yeah. All right, so we'll be going over the different testing methods that can be done within the systemic toxicity test, including how to tailor your test to make sure that it is most clinically accurate and representative and appropriate for your device type as well. And then also discussing a little bit on the types of observations and results and data that you can get from this test method.
And look forward to seeing you guys then. And well, I guess we won't see you, but you'll see us.
Yes.
But look forward to the discussion and happy digestion.
Hello and welcome to Bite-Sized Biocompatibility, the educational series focused on making the technical content of medical device biocompatibility digestible. My name is Leah Williams.
Hi, and I'm Beau Rollins with Intrinsic Medical Group, and we're excited to take you along on this journey.
All right, and hello and welcome back to Bite-Sized Biocompatibility, episode number 9, I believe we're on now. Yes, number 9. We've made it this far. Amazing. Today we are going to be talking about systemic toxicity, and honestly, this is one I'm really excited about because systemic toxicity for me is this type of study that you really get a look at everything. I think you also get that a little bit with irritation and sensitization as well, but with toxicity, you're looking at so many different organs and like clinical observations and different responses within the tissues as well, that I feel like it's a test that really does give you a lot of information.
It does, and unfortunately, it is an animal, right?
Yeah.
It's an in vivo test, but through the years of my career, the one test that, before obviously we got to chemical characterization and TRAs, but the one test that I generally would lean most of my evaluation and personal comfort level on was around the systemic toxicity test. Definitely not cytotox, as we've talked about, right? I'm not going to make determinations on that. But the systemic toxicity was one of the best ways to be able to determine whether or not the compounds and constituents coming off the device is going to cause a reaction.
Yeah, and I think what's good about this as well is that often when you're looking at your -18 and your -17, so your chemical characterization and your toxicological risk assessment, one of the biggest challenges that it faces is that synergistic effects of the different individual chemical constituents which may be exposed to the patient during clinical use. And so applying, this is typically an extract-based test, so applying that extract to an entire in vivo system, you can then therefore also account for those synergistic effects that might happen. And by synergistic effects, for those who aren't so familiar, it's those effects that can happen in conjunction with a couple of different chemistries that maybe on their own are fine, but like together they'll result in some sort of adverse harm.
No, definitely. And like all of the toxicological risk assessments that we spoke about in the previous episode are built off of systemic toxicity tests. They are with neat chemicals, so they are looking at individual chemicals and at what point do they cause a response, but they are all based off of animal toxicity studies. And so when we look at this for medical devices, this is one, as you've eloquently put it, that we can actually look at synergistic effects where we can see when we extract and have all these working together, do they elicit and cause a response? And generally speaking, in my career, there's been a couple times where through a TRA of the individual components and everything looked fine, but when we moved to an actual in vivo model, we've seen responses because of those different compounds working in tandem or in conjunction with each other.
I'm going to put you on the spot here and ask you if you have more details about a specific example about that. And if not, we can cut this bit out.
So, once upon a time, we were working on an absorbable material. And so with these, we know obviously that over time they break down. Have a full detailed list of how they're made, what's in there, also, chemical characterization as well. So we knew everything that was in there. And we did a toxicological risk assessment for each of those aspects in individually. And all were below levels of risk. Then when we stepped forward to actually do an injection study and an implantation study where they would then see this, and keep in mind, it was a worst case because we did digest or dissolve the absorbable, so that way it was a max dose, right? So that was the intent of the study, to be able to make sure that as a worst case, if everything was bioavailable at one time, what would we be doing? And unfortunately, when we did that, the ions that were in there was some calcium ions and phosphate, and that they worked together to weaken the blood-brain barrier and end up causing challenges with the animals. Would that happen clinically? As we looked more into the thresholds on humans and the amount we dosed and everything else, we still were okay on a human level, but it was a good reminder that we are approaching threshold levels that potentially could cause problems. So that meant that we have to look at the size of the absorbable material. How big can we make it before we reach that threshold?
Yeah.
And how fast can we allow it to dissolve or break down in the body before we have any sort of concentrations that could impact that?
Right.
There's always additional risks that factor in there. Like, you don't know the patient's diet. You don't know if they're taking supplements and things that can offset some of that other things. But it does help us at least know where we're at in regard to things. But so there's a lot of value that can come out of this test. There's always the kind of, been historically discussion, can we eliminate this test? As of today, like, we are through TRAs able to mitigate and step through generally executing this test for most markets and industries.
Right.
But I still personally think there's a little bit of a caution there that we don't always know how things work together.
Yeah.
And I'm not trying to downplay on the TRA or anything like that. It's just, there's so much complexities in our realm and things to
within
consider.
like human physiology as well. There's so many different pathways and metabolic mechanisms as well at play. You don't know, and I think that's kind of one of the main limitations behind that TRA and why it isn't just your one-stop shop for everything.
And it's important to note, like with systemic toxicity, any of the tests within this, and we'll get into on these. So when we say systemic toxicity, there's a variety of tests that kind of fall underneath here. When you get into -11, we talk about acute systemic, which is a single dose acute response that we're going to be reading or looking at the animal for 3 days, right? We have the subacute, subchronic, and chronic, and these all
don't question the names for them because they are a little counterintuitive.
They are a little counterintuitive, but more or less the subacute, we're looking for repeat dosing up to about 28 days, kind of around a prolonged period. When you're looking at subchronic, you're going to go past that 28 day, generally speaking, a lot of times up to 60 days. And then a chronic test, honestly, with medical devices, we generally have never performed these because of how long they take to.
I don't think I've ever seen a report for a chronic systemic toxicity study.
If you truly have something brand new and novel and it has potential reactivities, then maybe you could look at doing something like that. But I would say across med devices, very rarely that anyone has done a chronic toxicity.
Right. And so just to kind of like step it back briefly there as well, when we're actually executing the study, most typically or most commonly seen is that systemic injection study where you have an extract from that device and you apply it, well, as it suggests, systemically. And there's a few different routes that you can apply it by, for example, intravenous injection would be the most common one, but there's certain solvents that aren't compatible with that. For example, you wouldn't put your oil extract intravenously unless, I don't know, you want harm for the animal, which is not, let's not do that. We're scientists here. You'd just do that for your saline extract. But you can also inject it interperitoneally or subcutaneously as well, or a combination of them all if you are looking at those different different solvents.
One of the misconceptions with systemic toxicity is most people think that there's one test that we do. One test in each one, right? And the standard is actually written with a lot of flexibility and options that for you to tailor it to your device's intended use and need. But you're absolutely right. Everything that we do in med devices, we've expressed through multiple episodes, is worst case, which is why the standard approach is to do an intravenous injection with your polar and then intraperitoneal with your non-polar.
And this is considered the highest risk because systemically it's whatever chemistries you've got are transmitted the quickest throughout the body.
Correct. It's directly circulated throughout the entire body all in one dose. And then the other aspect on this that goes above and beyond is the volume that we inject in there. So I think
a lot of people don't realize just how great that dosing volume is, especially if you're dosing in, say, your typical models like a mouse or a rat model.
We're not talking like 1 mil. We're talking upwards to 50 mils at times on injections. So much that it's so much fluid that you do it slowly over a period of minutes to be able to allow the body to just absorb and try to handle the volume itself.
I'm not quite sure it's 50 mil, by the way. That's quite high, but it's in the range of milliliters for sure.
Well, it's mils per kilogram.
Gram of body. Per kilogram of body. But for the rat study that I observed in Ohio, that was like 1.4 mil.
Correct.
So for a rat.
Because it's super light.
Yeah.
But it is 50 mils per kilogram.
Gotcha. 0.5 milligram.
Yeah.
All right. Glad we got that clarified. I was like, it sounded like you were saying we inject 50 mil into a rat. And I was just like, oh.
Yeah. Good clarification.
Don't do that.
when you look at the table on the dosing levels and that, it is, listed in mils per the animal, but it is per body weight. So it is going to depend on the body weight of the animal.
It's still a very high dose.
It is still extremely high dose. And so much that when we inject like the oils, right, a lot of times you'll get a pocket formed and you have to sometimes they massage to try to get the oil to disseminate, or they have to repeat or extend the injection times over like an hour or more to be able to allow the absorption of it. So the load volume that we do is astronomical. So when you already take that in consideration from a worst case, that's above and beyond. And that goes with every dose route. So it doesn't matter if it's intravenous, subcutaneous, or anything like that. The dosing amount doesn't necessarily change. And then, yes, you want to look at clinically relevant dosing, although the standard test, if I went to a lab and said, hey, I need a subacute test, they're going to set up an intravenous injection.
And I think that's actually really good to kind of bring that back to that clinically relevant as well, because, say, for example, that you have a wound dressing or a surface device and it is used for a long-term duration through cumulative repeated use. But devices like wound dressings don't really lend themselves very well to extraction. For example, if you've got a wound dressing that can absorb a high quantity of fluid, you may get a lot of degradants and particulates coming off with that as well. Try and inject that intravenously, you're going to run into a whole host of issues. So actually, this segues quite nicely. What other maybe not quite as conventional methods of dosing are there other than just an extract-based method?
So there are some custom protocols out there that actually do application. So that applies a lot more to breech skin devices where they'll actually create a defect with the animal. This is done on small models like a rat oftentimes, but you can even extend it to a large model. The
like
standard allows all that flexibility, but you have to keep in mind that it's not a wound healing study in this sense. This is a toxicity study. So ensuring that you're getting the appropriate dosing compared to clinical use and repeat application while also maintaining that exposure route.
Right. And I think this is also where it's really important to kind of maintain that wording correctly, work with your study director to make sure that wording in your resulting report is also saying toxicity study or systemic toxicity rather than wound healing study, because I have seen toxicity studies where they kind of get a little bit confused because a regulator sees it as like, oh, well, you administered the dosing through a wound, therefore it's a wound healing study. And even like within your manufacturing teams as well, they see it as a wound healing study. They try and use that and apply it for different endpoints that clinical teams have to use as well,
Yeah.
looking at wound healing, but that's not the intent because we're looking at that exposure to the systemic toxicity exposure of that chemistry.
Yeah. And when we get to implantation, we'll talk more about wound healing studies because that is where that appropriately falls under is -6. But it is important to note that there is a bridge between -11 and -6 that allows you to combine these studies. But you need to be very cautious on combining these because most of the time we combine, it's still an implantation based off its acceptance and requirements and not fulfilling a toxicity study. Very rarely do you see a true toxicity study utilized through an implantation. It takes dedicated animals a lot more, and then the dosing aspect to encompass as part of the study, where most implants are going to be a one-time dose or exposure, right? And you combine oftentimes the animals with controls or with your predicate device. So we'll discuss more on the implantation side of things when we get there, but for the toxicity side, there's all sorts of dosing, right? And when you're talking about like foreign particulates and things that come into play, this is very critical things to watch out for, especially if it's not part of clinical use, because we are extracting devices and things. Particulates can cause a lot of problems. You can't just filter them generally unacceptable manner. If you do filter, you need to go through a bunch of analytical work to show that you're not filtering out the constituents and compounds, but you're just removing the physical and what they are, so identifications of those as well, so that way it can be clearly shown that this isn't part of clinical use, but we have removed them. So that there's a lot of complexity that can step into this test. This is not just an easy slam dunk type test that you can.
There seems to be a bit of a reoccurring theme in biocompatibility. And again, it's just about really making sure that you're painting that full picture of that risk evaluation. There's not just one piece to it. There's multiple parts to the puzzle as well. I think just like bouncing off the back of that. So another way that you don't do the exposure through just an extract-based test, or another situation where you wouldn't just have an extract-based exposure would be, say, if your device is a gel or something, and that can actually be injected subcutaneously as well.
And with all this, keep in mind that following the standard doesn't always equate right to regulatory acceptance. I've personally seen where I've taken a device that clinically is intact mucosal, and having the agency request a 14-day, 14-dose IV, injection study. So even though that's not at all clinically relevant, going back to execute that, and then matters of months later, going with another device, very similar device, just different configuration, and having a different review team come back and say, we don't want a 14-day, 14-dose because that's not appropriate.
Not clinically relevant.
Not clinically relevant. We want a different dosing. I've also had them respond on similar device on subcutaneous injections asking, how do I know that it's bioavailable when administered through subcutaneous or injections?
Well,
so,
the answer is technically you don't, but you're making it more bioavailable than it would be clinically.
Yeah, well, I mean, if subcutaneous injections didn't work, then diabetics would be in a world of hurt because all of insulin's administered subcutaneously. So it's something to be aware of, like when you step with this test, is that you never know what kind of questions you're going to get. So it needs to be very thought through and planned through the stages early on. We talked about feasibilities when we mentioned in -12 and how important that is for extractions. This is also extremely important to be able to do feasibilities here. And when we talk about feasibilities with systemic toxicity, it's very important to note that this should be done with any device that you have any sort of soft materials, weak materials that are impacted by extractions and degradation, because you don't want to set up a study with 10 animals or more animals and then have them all die because of incompatibility with the extraction. So there actually is single animal feasibility that you can set up with your CRO where you can verify the extraction aspects, especially if you do see discoloration, particulates, or any degradation of your device.
I've even seen it before where you do get an extract which looks like it should be suitable. You've done so another couple of checks you can do is check the pH of the extract prior to injection. You can check the osmolality as well if the test lab that you're using has that capability. And usually if those are within range, and you can still inject it with an animal and see adverse responses, there's a lot that could go wrong with this test. So doing that feasibility and really is important, not just for the success or directing how you're actually going to go about conducting that method, but also it supports in that justification of the parameters that you use within the methodology too.
Yeah. And unfortunately, sometimes you have to do the feasibility to prove that you can't do the test.
Yeah.
And instead of trying to execute a large-scale model where many animals are going to unfortunately die from that, we want to minimize that impact. So therefore, do a feasibility first to show that it doesn't work, and that then helps with the justifications.
A question that just popped into my head there. So can you then, there are obviously number of animal requirements for these studies as well. Look to the infographic for more information on that. Could you then say, if you had a successful feasibility in the methodology that you wanted to progress with, you're like, right, this is good, we can continue into the full scale study. Could you then use that data from the feasibility to be a part of that, meeting those animal number requirements, and therefore use fewer animals?
Unfortunately, no.
Damn it.
Yeah. Unfortunately, because the feasibility isn't going to be set with the same animals acclimated at the same time and having the appropriate numbers, you're looking at most likely different extraction times, different extraction, even control solutions. We usually, when you order in a test, they'll get a group of animals. They have to have them together to show for a period of time that they're safe and stable. And then you do an extraction set together, right? So you're getting all of those extractions at the same time.
It's about reducing those variables, doing good science.
Can you technically, per the standard, split that out? Technically, yes. There's nowhere in the standard that says these all must be extracted and administered at the same time.
Yeah.
So you do have the ability to break this out, but keep in mind that anytime you're starting to piecemeal, right, your reports are going to look messier and you're going to get more questions.
Right. So best if you are going to do that approach, balance it out with equal numbers of animals. Say, for example, if you have a large scale study that has multiple animals and it's actually going to be really logistically difficult for the lab to execute that many, just make sure that it's as balanced as possible.
Yes.
And split as little as possible as well.
Yeah. Unfortunately, anytime you step to a large scale model like a canine, a sheep, or porcine, most labs will only have 1 or 2 surgical suites that can, or staff vets that can, execute these tests. And just due to the volume of animals that's needed for all of the points, it's impossible to do this all as like one day type execution. So you will be looking at setting these up and spreading them out, especially if they don't have the husbandry facilities to be able to maintain the animals, especially if you're doing like a 45-day study or 60-day study. So you very well might have to set up multiple groups that go through. But the importance there is obviously make sure that all your test articles, predicates, and controls are all the same lot and batch. They'll just have to be held and maintained and used at different times.
Again, just minimizing those variables as much as possible, controlling what you can.
So many details that go into these studies. They are not cookie cutter assays. I mean, that would be my tip and recommendation to everyone out there, is that this is not a cookie cutter test.
No.
Don't be I don't want to say fooled, because I don't think the CROs are trying to trick anyone.
No.
They're trying to just standardize things as much as possible for them, so that way they don't get errors and things.
Right. Well, it's also better for human error as well. It reduces that uncertainty. They know then what they're doing. They're feeling more comfortable as well. But actually, that's a really valid point there. Think about, as with all biocompatibility testing, but especially with sort of systemic toxicity testing, really think about how is best to set it up for your device and the device use, and how it's going to best represent that clinical exposure seen during device use as well.
And then as far as tips for people who are first time executing this type of test, obviously how it's extracted and dosing needs to be considered, and make sure it's appropriate for the device and the clinical use. Yes, well considered. Keep an eye on, and I highly recommend that you go for the administration phase of this. That way you can better understand how it's being done to the animal. You can also observe. The first stage of this is going to be clinical observations. So
and just before we jump right into that, I just want to kind of really reiterate that point as well, and say that actually CROs are very happy to have you. In fact, they'd rather you be on site as well. And from experiences where we've both been on site with a CRO for these types of studies, it's really useful for the lab teams there as well. If they have any questions, if they see something and suddenly, so there's so much more than just what's written in the standard that goes behind the methodology, and it's really invaluable to go have a look and just see kind of all the considerations that go into it.
Very good point, because you know your device. The CROs do not. And even though we think our device is straightforward, I've had many instances where the wrong pieces are extracted or cut or missed because they don't understand the device, and that's not their fault.
And a good way around this as well is if you can't be there's a number of surgical suites that do have a live stream option as well, so you can watch in remotely. And then always ask the CROs to share any photos with you if possible as well of the device that they extracted, because if they're conducting it under GLP, so good lab practices, they will be taking photos with all of it as well.
And I know animal testing is always a sensitive topic, and there are a lot of people out there that are genuinely, disturbed and affected by this, and
it's hard to watch.
It is, and being aware that this occurs. However, out of respect to the animals, it's, from my view, it's more important that you be there to observe to make sure it's done correctly, so it doesn't have to be redone. Because nothing's worse than there be a mistake that's done through handling, extraction, or administration that then requires you to have to repeat the test and impact more animals.
Get it right first time, right?
It's a hard truth and reality of our industry and what we have to do for patient safety, but own that and make sure that you minimize the animals that are implicated by making sure it's done right the first time.
With the sacrifice of the animal life, make sure that the data is going to be actually used to its max, and make sure that you get all of the data that you can out of it. And that segues nicely actually into
the clinical observation
the clinical
aspect.
observations and the different results you get out of a systemic toxicity test.
So when you conduct a systemic toxicity, there's really kind of two sets of data, is how I like to explain it. So we have our clinical observations. This is going to be what's recorded and observed during the testing. So once administration is done, or prior to administration, the animals are weighed. They're also observed and recorded, looking for set reactions. So they're
like in their behaviors,
right? If they're having any sort of tremoring or shaking, if they're not eating, if they're not drinking.
They even look around they've got like animal pain scales as well that you see in the photos of the labs as well, where they look for responses about how much they're squinting in terms of their eyes and all of that. So if they're socializing, if they're moving around and generally looking happy.
So there's a lot to observe through this aspect. And so after the administration, especially if you're doing repeat studies, it's very important. The lab's going to be going in every day to make these observations, to record these things. And you get characteristics with the animals. You'll get some that just naturally have more of a impact from this, are more shy, or don't eat as well. Like you said, you get some that show pain, some that are scared, and observing these things through, looking for individual characteristics that are part of their personality versus as actually from your product is sometimes a challenge. But all this data is very important, and knowing and even being part of some of these reads, it helps you understand. So that way, when the data comes back and they're looking over all of these days of data, whether or not to ascertain that the response is observed, if that's actually due to the device or due to the personality. Then at the termination phase, that's then what everyone normally thinks about with these studies is that, we're going to get our histopathology, we're going to look at the organs and the tissues, and we're going to look at how those have been affected by the administrations. Unless you see gross catastrophic responses, you generally aren't going to see a whole lot through, in my experience, on these, because we're not using very reactive materials in that.
No, exactly. It all goes back down to that design. We don't design our medical devices with those harmful substances, so typically you don't see a lot. And actually, so that kind of segues a little bit nicely into also the other types of observation that we get with this as well, is that before we jump into that histopathology stage, we also have a look at the necropsy stage as well, where all of the different organs are evaluated in terms of like their weights, comparing it to kind of the average weight of each organ. There's also a macroscopic observation that's performed, looking for maybe discoloration of tissues, if there's any formation of nodules on certain key target tissues as well. And I think another aspect as well that can be another type of data that you can get from these studies as well, which maybe isn't so routinely done within the systemic toxicity, is the sampling of the tissues during the study as well. So looking at maybe blood samples and the biochemistry within that, and urine samples as well.
Yeah, that's often, as far as people should know, that's not standard protocol.
No, you need to request that if there's something specific that you want to be looking for as well. Usually the labs are very flexible if you say, hey, I want to also have an investigation into this aspect.
But make those decisions based off of what data you need. So if you're looking at having like a PVC stent or tube that's placed in that has no coating or anything else, do you really need to look at urine and things like that? No.
Exactly. And this is where the chemical characterization piece, yet again, feeds right into it. Once you've done that data gathering stage, you know what the constituent materials are. You know roughly what risks you're going to be screening for as well. So therefore, make sure that your study design is tailored to look for that. So instead of just checkboxing, really identify where those gaps are within your evaluation or where more information is needed, and make sure that your study design is tailored for that.
Yep. Couldn't have said it better. So it's, as you can see, there's so much that plays into this, and there's a lot of good data, but there's also a lot of data that can throw confusion. As you mentioned, nodules and things on organs and stuff, which can be genetic diseases from the animals itself. And there's times when you'll see one or two of your test articles, and that's the challenge here, is if you don't see it in control, you don't always know if it's from your device or not, even though you might know that those nodules on the liver are very much a genetic disease. If they're not found on the controls, then it causes complications in the results.
Well, equally, there's also been a few test reports that I've pulled up, and I nearly have a heart attack seeing some of these tissue observations, and I see that they're also seen in the control animal. I'm like, okay, good. Yes. It's just a genetic thing.
I was definitely going to say, when you start reading through your pathology reports and stuff, like just prepare yourself. There's never a clean slate. You're always going to be observing different responses and different things. Keep in mind the trauma that the animals have gone through and the distress that they go through. All that plays on their system. And so, yes, you will find in your test article, you'll see like enlarged testicles, and then you start to panic, but then you look at the control and they also had enlarged testicles, and you're like, why did we have enlarged? It doesn't make any sense.
So it happens. But we'll be going into kind of like the different clinical observations, the necropsy, histopathology, and other tissue sampling in a later episode as well. So make sure you tune in for that as well. But I think that's about all we've got time for today. So thank you very much for that discussion. I hope this made systemic toxicity a little bit more digestible for you. And feel free to reach out to myself or Beau anytime if you have any questions. You can contact us via LinkedIn, or we'll drop our email in the show notes as well. So, come ready for the next time. We look forward to seeing you.
See you then.